Some plants have unique defense mechanisms which are triggered in response to a particular threat, such as insects trying to feed on them. For example, plants such as soybean sense herbivore danger signals, or HDS, specific chemicals in oral secretions of insects. But how do the plants recognize these herbivore-specific elicitors? Despite decades of research, this has remained unclear until now. In a new study, researchers from Tokyo University of Science attempted to provide insights into how plant HDS systems work. Based on previous studies on model plants like Arabidopsis, tobacco, and cowpea, they focused on membrane proteins called receptor-like kinases, RLKs, which play a major role in HDS systems. To begin with, the scientists focused on RLK genes in a soybean plant that were structurally and functionally similar to an Arabidopsis RLK gene, known to trigger a danger response by recognizing small carbohydrate molecules called polysaccharides. Through genetic analysis, they found 15 such genes, using which they created 15 types of transgenic Arabidopsis plants, each plant uniquely expressing only one of the individual soy genes. Then, they tested these plants using oral secretions from an insect. Interestingly, they uncovered genes for 2 novel RLKs that showed a defense response specific to the oral secretions, called GMHAK1 and GMHAK2. Moreover, when the scientists dug deeper into the mechanism of these regulatory factors, they found that 2 proteins playing a role in intracellular signaling in Arabidopsis, ATHAK1 and ATPBL27, are involved in this pathway. These findings confirmed what the scientists had initially expected. Soybean and Arabidopsis possess similar mechanisms for a danger response. This study is the first to show that in Arabidopsis plants, HAKs and PBL27 act in tandem in response to HDS. This insight into plant defense strategies could potentially fuel various developments in agriculture.